Hey, I'm Dontrell Sultan, the host of the Innovators of BC podcast. And the Burroughs School of Business and Entrepreneurship is where bosses are built and big ideas take flight. Welcome back to the Innovators of BC podcast, powered by the Best Innovation Lab. I'm your host, Dontrell Sultan, with our guest today, Mr. Robert Hamilton, and he's a junior biology major, and we basically wanted to bring him on this podcast to talk about his research. And this is a little different than what we normally do, but I thought it was really dope for what his presentation he did on Research Day. And actually, didn't you get a prize for that? Or anything?
Yes, that's
Really?
correct.
So that's how I know it's credible, and it's basically the research on what fertilizers do to plants and how mutated plants resist those fertilizers, right?
Yeah.
Well, that's my simple term, but obviously he'll break it down in way way better terms. But how you feeling today, Robert?
I'm feeling good. I'm feeling good.
Really?
Yeah.
You coming from class?
Yeah.
What class was it?
Probability and statistics with NAID.
Why do you have you taking that? Is that like generated?
It's required.
Oh, like I don't understand. Why is that required for biology majors?
I think it has to do with the whole the general population mechanics in like the latter half of the class. But
Uh,
it's like pretty much it's just a bunch of bio majors, senior and junior bio majors in that
uh,
class.
well, that makes sense. But we had business stats, so it was more operations and looking at population sample size or whatever. But yeah. So talk about where are you from?
Jamaica,
Which part?
Portland,
Portland,
Jamaica.
Jamaica. Okay. Hey, I know a couple of people from Portland, Jamaica.
Yeah, I think we all came to school.
Did you come on like a scholarship?
Yes.
Because I was gonna ask, how did you discover Benedict and how has your journey been so far at Benedict?
Well, I discovered Benedict because like a few of my friends from high school
Mm,
came up here and like they obviously talked about the school that they went to and I was just like, well, Benedict sounds like a great option. So I applied. I got a pretty good scholarship.
mm,
Um, Even though I kind of came up here a bit late in the school year.
mm.
I came like after like I think in the third week of school.
Third week of school was it because airplane?
Yeah, it was like
Okay, yeah,
probably
yeah,
shifts or
travel.
but that's my journey up here. It was pretty decent. Like at first it was a like, culture shock.
Mm,
But I acclimated pretty quickly, and ever since that, like in the freshman year, I've just been
mm.
pretty good.
So why not? Well, I don't. Did Jamaica have any schools that you were interested in? Like I'm saying, I'm wondering what differentiated that those opportunities for
Um,
you.
well, with my other options, back home, like with the University of West Indies and like UTech, there was a want to go there because there was like a very like rich history there, and like I had family that went there.
Mm,
But what ultimately made my decision was just like I just wanted something new,
mm,
and I just wanted like opportunities that were up here were like more not enticing but important to me.
mm.
It was like very important. My family especially like really pushed. My father really pushed for me to like go to a school in America or go to a school in abroad. Like he wanted me to have that experience.
And have you always been fascinated in science?
Yeah. Yeah, it's hard not to be. Like my mother did like environmental science. My father is a physical education and physiology person.
Wow.
So like I've been like since I was four, just like reading biology textbook, reading chemistry textbooks. It's just always been like a part of my life.
Mm. And what kind of been a pivotal project or piece of research that kind of really instilled that passion or that interest?
I think is what when I was like in primary school, which I think is like the equivalent of like middle school
Middle school.
here. It was when I was like in primary school, and I don't remember all the details, but I do remember the research. It was a study on to see the growth of like algae and rocks within a nearby drain.
Mm,
And it was just like going over there like every day for like a couple months, just like measuring and like, taking data and collecting samples and like going back to that little makeshift lab that they had in the school and to just like, do some tests and like run some fun experiments. And it was just like, oh, like this is what I want to be doing.
mm.
Like this is like it for me.
That's dope. And talk about now at Benedict. What kind of drives that passion and keeps it flowing with your major? Has there been pretty cool projects within your majors that you want to highlight?
Like a few like projects that I've like done is like obvious my research with Surrey
Mm,
and my research that I've done with like my professors in my chemistry classes and my human anatomy courses.
mm,
A lot of the work that I do like pertains to a lot of like things that I'm doing in my courses.
mm,
So it'd be like service learning projects with the school to do things like cleanups and like synthesize chemicals to help with the cleanup, synthesize things like alums to
mm,
help with like cleaning water sources, for like things like organic chemistry, or in the case of human anatomy and comparative anatomy, would be projects that have to do with like identifying bone species of gar. I can identify their bones, identifying
mm.
like specifically their subspecies and helping with categorizing them, like things like that.
Okay. And let's get into your actual research that we discovered you on. In the beginning, I probably butchered the explanation of what your research is, but I wanted you to elaborate. What was your hypothesis and what was the goal of this research?
Well, the hypothesis that I came in with was that I had this idea of like researching the effects of over-nitrification because I grew up in an area that was like pretty rural, and my family was like, again, my mother was environmental science because her family was like, we lived on a farm.
Mm,
So I kind of was like familiar with the concept of like over nitrification and like the concept of like over polluting soil, like wash off and detergent, because things like that tend to happen in like areas like that. So I was like, my initial thing was looking at over nitrification of soil and whether or not that affects plant growth, because I knew it affects plant germination,
mm,
but what about like plant growth? So my initial hypothesis was that like if you use too much nitrogen-based compounds within these enzymes, acid phosphatase, which is like the enzymes responsible for the production of phosphatase, and any biomeasure will tell us that that's important, like ATP,
mm.
which basically fuels growth and development. Um, but what I wanted to see specifically is like inhibited these enzymes specifically. So I think it was not freshman year, summer. I took it upon myself to just like work under Dr. Lowe, who was working with the Unpack project, who kind of like introduced me, said, oh, you can make it specifically about this project.
Mm.
You could work with these like the Arabidopsis plant family members, which Arabidopsis plants are just the most, includes like some of the most common like vegetables, like broccolis, kales, cabbages, lettuce.
Mm.
So it's really like grown for agriculture type stuff. And I kind of just like when it's like, okay, then I'm just gonna introduce too much nitrogen into these things and see whether or not like is the enzyme activity working or not, essentially. And then they kind of grew with like the introduction of the mutant plants and everything.
Mm. And you said these plants were the basic vegetation that we eat.
Yeah, like it was like specifically chosen to be that because it's like this is like some of the most common like grown crops in all parts of the world, can be grown like almost anywhere,
Mm.
even in like certain of these plant members can be grown in like even extreme conditions. And their wild variants were like everywhere, like even in like common gardens.
So looking at a farmer, does this benefit the farmer as far as like I can produce it at a mass or I could produce more due to this mutation? Or is it the plants will be able to last longer in extreme conditions? Is that why these mutated plants are beneficial or harming them?
Um, in terms of like agriculture and like benefiting like a theoretical farmer, would be more like these plants could withstand like poor soil quality or like soil that was like had a problem with like the nitrogen cycle in it, like soil that previously was either like over fertilized, so like it doesn't really have as much of the other nutrients in it, and like the nitrogen could like choke up like certain phloem structures within the plant. Like these like mutant plants would have the ability just to resist that and grow despite that.
Hmm.
So you would not like you could like focus your time on like trying to fix the soil rather than being like, oh, my crops are dying. I'm gonna have to like wait to fix the soil
Mm.
and then grow. You could just like grow those plants and then, fix the soil. So if you can want it to continue and just like get a bigger yield next time,
Mm.
like these plants would be there and be able to resist and like wouldn't like, die or would you produce a smaller yield. Like these plants would like be able to like resist that.
Wow. And how does this vegetation affect us?
Well, further research needs to be done with that, but these mutant plants seem to only have mutations pertaining to the enzyme.
Mm.
In terms of their plant growth, since I grew the generations myself, there was any phenotypical change or difference that I could see within these mutant plant samples. There was no like change in coloration really.
Mm.
There's no change in like growth structure or how they grew. Just
Mm.
that they, in the presence of this nitrogen, they were still growing
Oh.
compared to like the non-mutant samples that were like starting to die or like didn't even get the chance to like grow past like a seedling.
Mm. So did that hit on, well, you said you would have to do further research on that. So doing further research, is that one of your points that you want to kind of meet, or do you have a different purpose as far as future research? If someone was to invest into this.
As far as future research, I would like to see like broader application for different artificial family members. I would specifically want to like single out what genetic mutation
Mm.
affected the enzyme in this way, or is it just like a generalized, oh, once you change the enzyme in total with like any gene change,
Mm.
it just like develops this resistance automatically as like an immune response, or to like just specifically like see if this can be replicated in further generations. So like growing more generations of the mutant plants to see, all right, then does this mutation randomly change? Does it have any side effects over time?
Mm.
Does it like cause it to be like less resistant to something else?
Mm.
I would also like to test it on like more commercial, like members of the Ardossus plant family, because the genus that was used was specifically like a genus that was eaten, but it was more like a like wild
When you say
version of it,
commercial, you mean
like
use,
with kale and lettuce and
okay.
like broccoli,
Mm.
like mutated versions of those plants to see if like this genetic mutation is transferable to like those plants, and you could like breed hybrids or breed like mutant versions of these plants that are like more resistant to this, and whether or not those are safe for consumption or like they have any sort of like side effects like affect the taste or
Mm.
affect like specific nutrients like component it, things like that, things in the realm of food science.
And what do you think that would require as far as equipment, and because what were all the methods that you used for this research?
The methods that I used for this research was, firstly, I had to like specifically find out the amount of enzyme within these samples, the non-mutant and the mutant ones, which was like a simple protein assay,
Mm.
and with the samples just to get like a standard curve so I can work off of. But the real meat of the research was utilizing PNPP, phenyl nitro fluffinate, which specifically was just like an indicator that like this showed that the enzymes were working because it basically changed the color of the compound yellow
Mm.
when in the presence of phosphorus. And these enzymes produce phosphorus.
Mm.
So I was just doing like a simple test to see enzyme activity. I wasn't like specifically doing like genetic work or seeing like, specifying like, using things like CRISPR technology to see specifically which tools, like which like series of genes, like, which series of like DNA and RNA like make up the mutations. I was just like testing to see if like these enzymes were working.
Mm.
So I just used a photospectrometer and like tested, well, it's gonna turn more yellow the more phosphorus there is, and like you get like a good reading as like then it turning yellow over time. And like the changes in color indicate the changes in like enzyme activity, which because it can like indicate the changes in like how much phosphorus is being produced over time, which led me to see like, oh, so when without the an example would be like the Tween 20 like detergent,
Mm.
it like it gives you like a standard curve, like you see it working, and over time it kind of plateaus because there's like it's already producing enough. But with the Teen 20 you can see that like it barely starts to work and then it like instantly plateaus.
So as a consumer, just beyond this research, what type of, whether that's nutritional facts or just a specific brand or commercial vegetation. What do you think we have to look out for when we're shopping for this vegetation? Like, do we have to look out for anything in those nutritional facts or anything like that?
Like it that kind of gets into the talk about like GMO and
Mm-hmm.
GMO foods, where it's just like a lot of GMO foods and a lot of the plants that we consume are heavily genetically modified through whether it be breeding or specific genetic transfer, like with the mutant plants
Mm-hmm.
that I use in my my research. And it just boils down to the fact that like these plants, like no matter whether you like not really have an understanding of how GMO stuff works, is that you kind of have to understand like this is done in order to increase nutritional content, in order to increase flavor, in order to increase resistance.
Mm-hmm.
What you need to look out for specifically, I would say, isn't necessarily fully related to the mutations in those plants, but more so the shelf life of those plants. Because a lot of these like GMOs and like a lot of these things do extend shelf life, but
Mm-hmm.
only under specific conditions. So like the non-mutant version of the plants I use had a like short shelf life, which is like the expected of like a vegetable like a broccoli or like kale. But the
Mm-hmm.
mutant versions I saw like really lasted longer outside of like their usual environments. Like I didn't have to refrigerate them or keep them at like temperature controlled
Mm-hmm.
for them to like last as long. So like just look out for things like shelf lives, be aware of like how long can you keep this before like it becomes spoiled, and be aware of like specific things like allergies because a lot of GMO stuff can like trigger certain allergies and immune responses.
Really?
Yeah, like surprisingly, it's like one of those things where it's just like your body isn't used to like other foreign stuff in it. Sometimes it just like
Mm-hmm.
attacks it without
It,
cause or reason.
does that trigger the allergy for eating that type of plant, or would it trigger some additional allergies outside of that?
No,
Oh no.
just like if you already are, it could be get worse.
Okay. And with these, you said long lasting shelf life, right? So that obviously that looks abnormal to us. Like, okay, naturally it's supposed to last X amount of time, a small amount of time, but now the GMOs was allowing it to last for a longer amount of days. We hear that is abnormal, but what can you say that this abnormal long lasting shelf life really affect our health?
In terms of health,
It doesn't.
not necessarily. Like there needs to be more research done into that. But like if we're using like the template or like the history of like other GMO foods,
Mm.
really and truly there isn't any like long term negative like lasting effects on like health.
Instead of
Like
allergies.
yeah,
You're
like
right.
we have like as in agriculture for like centuries, if not millennia, like modified our foods. Like with members of the Arabidopsis family, a
Mm.
lot of them initially came from the same plant. It was just like selective, like culling and breeding through the generations, kind of like diversified the species like forcefully.
Mm.
So like this is something that people have been doing for as long as they have been like farming. So there is like if there was any negative impact on health, you'd have like known by now.
Really.
Like we'd have like seen it in the human population by now. Like things like Brussels sprouts, cabbages, broccolis are all just like exaggerations of different parts of like a similar plant like years ago.
Mm.
Like you can see based on like how they grow with like broccolis being modified flowers, cabbages and Brussels sprouts being modified leaves,
Mm.
kales being modified like buds, and like celeries being modified stalks. Like this is just like something that people have been doing, like farmers have been doing. Like even back like home, like my grandfather used to have a thing in which where like he specifically grow like yams a certain way to like make sure that they were like larger than like other people's yams. Or like he'd grow like them in certain ways to like to make sure that they were like softer when they were like being picked, or like they'd be like easier to identify as like his. This is just like tricks that you learn.
But I feel like always in history, like if you decide to poke with something and modify something, I feel like it's some type of negative effect, like whether that's maybe I don't know cancerous or something like something, or you're more prone to maybe be gain more weight out of it or get more. Well, obviously because it's more nutrition and it's more. So I'm saying it's got to be some type of negative. like reasons because of it. You know what I'm saying?
Yeah, I understand that concern from like a lot of people. When it comes to like a lot of like the understanding, like people have been like doing stuff like this for like generations, knowingly or unknowingly, kind of like always selecting for like the most, I wouldn't say best or advantageous because that does not give a accurate view. Like the most convenient
Mm.
version of something or the most nutritious version of something. Even like things like bananas or like watermelons, like a lot of these like fruits don't grow necessarily just for the want of to feed other people. They want to like spread seeds. We just like have chosen fruits that like produce more food
Mm.
and smaller seeds, like with things like bananas and such.
Man.
It's just like there isn't really like a thing like if there is like a carcinogenic like there more cancer or more weight that would require like human testing and human trials and like I'm not familiar with food science as much as I'm familiar with like medical research and botanical research.
Mm.
But like that type of stuff is like controlled by like organizations and bodies to like make sure like that stuff is like locked down.
Okay. And we're approaching our last piece of the podcast where we want to give the audience, normally it's a piece of advice for starting a business, but say it's someone, a young aspiring biologist or a kid that has some groundbreaking research, what can you give with as far as executing it? Like maybe they don't have the resources to execute it. So what type of advice can you leave us with for the audience?
For any like aspiring researcher or just like someone who wants to take it upon themselves, like get in the world of science, I would say like always make sure that you're looking at like what's going on in that field concurrently. Always make sure that you're reading articles. Always make sure that you're like you can parse information from these fields. And as for getting help with research, I would say like there is so many resources. Like the field of academia, especially in research in like biology, feels like it's closed off, but there's so many people out there who's like more than willing to hear your hypothesis, more than willing to help you. If you like go to local universities, libraries, there's like environmentalist clubs. There's like things like 4-H that will help you out there and will like give you a space, give you equipment, and help you along with your work. And then like a big piece of advice as well, your hypothesis can be wrong. It's so much like it's like a lot of people worry about whether or not their hypothesis was wrong. Like I worried whether my hypothesis was wrong like
Mm.
initially. Well, that's a part of the fun, kind of like proving whether you were right or not. And even if you're wrong, there's still research to be done to find out why.
Mm. That's great. That's amazing. I wonder. Let's say if it's wrong, but it wastes so much money like with researching it though.
You still find out something, you find out you were
Yeah.
wrong. Like there's that's the
Yeah.
fun part about research. You're always gonna learn something no matter what happens.
Yeah.
Even if nothing happens, you learn that nothing happens.
That's real. Don't take well life so seriously for real. That's real. And can you leave us off with your socials or the way for them to reach you with regards to this research or whether or not you want to continue it, like any type of social handles?
I don't necessarily have like social media for my research,
Would
but
you have what an email?
email Robert Hamilton, Robert.Hamilton at 69 at mybenedict.edu.
Okay.
If you want to ask me about research, you can always find me in alumni, or you can always find me at BC Cares with the honors program and with the HCAS team.
Mm.
Um, so if you want to contact me there, you can contact me there.
Okay. Thank you for coming on this podcast. We really appreciate you.
Thank you for having me on.
What's up? It's your host, Dontrell Sultan, for the Innovators of BT podcast. Level up your hustle at Best Innovation Lab, where the Benedict's boldest ideas come alive. Good.